hello everybody. My name is Cynthia Webster. I am from the University of Connecticut. I'm here with Jill. And this is a project I've been working on recently with NC State. And it's an expression analysis of needle abscission in Eastern North American firs. So first, I want to define post-harvest needle abscission, and that is a physiological process of needle separation from the main body of the tree after harvest. And needle abscission is often a selective criteria that customers use to purchase a real Christmas tree. And if we learned anything from Charlie Brown, there seems to be a correlation with a good needle retaining tree and having friends. So how do we grow the perfect Christmas tree? So we utilize an RNA-seq analysis to conduct this experiment, and we identified genes expressed at the extremes of needle abscission retention, so good versus bad, and 3 fir species used for Christmas trees. We heard of Fraser fir earlier, but we also worked with balsam and Canaan. So I think samples were collected when I was a freshman in high school. I have since graduated from college, but we extracted RNA from the needle abscission zone, which is where the needle meets the twig. And we did this in, again, good and bad phenotype firs. And these libraries were sequenced via Illumina HiSeq 2500. And just a note, the samples were collected on landscape across 2 years, so we have clones. And to begin our actual RNA-seq workflow, the first thing I did was I trimmed our raw reads, and I used sickle to do that. And then I used a multi-assembler called Oyster River Protocol, and we're left with millions of transcripts, and that's not good. We want to filter that down. So the next step that we'll take is isolating our coding regions with transdecoder, and that cuts the transcript number in half. And then we're going to collapse our redundant transcripts into one representative with V-search. Again, we have fewer transcripts, and finally remove contaminants and annotate with NTAP. So overall, our transcriptomes were roughly 150 to 250,000 transcripts. And we could assess the completeness of our transcriptome with Busco. And it was pretty good. It was between 97 and 99% complete. So moving on, upon gaining abundance estimates with Callisto, I could then perform a differential expression analysis on all 3 fir species. And again, we're looking at good versus bad condition. And I considered a p-adjusted less than 0.1 and a log2 fold change greater than or less than 0 to be significant for each individual fir. And that left us with roughly 500 to 600 differentially expressed genes. But we also did something kind of unconventional where we utilized a more lenient p-adjusted value, less than 0.25. And we did this for an among taxa comparison. So because we could not use a single reference for our differential expression analysis, and we really wanted to know the shared genes, by lowering our confidence and looking at only the shared genes, we're kind of assuming that it's a higher confidence if there's 3 levels of support. That was Ross's idea, but moving forward. So using this p-adjusted less than 0.25, now we're asking the question, do differentially expressed genes fall into orthogroups? And yes, they do. Almost 100% of them do. So if we visualize that, we're going to look at this Venn diagram. And this is just representing the number of orthogroups with D genes that are shared and unique between species. And if we want to figure out what differentially expressed genes are shared by all 3 species, we're looking right in the middle at the 40 orthogroups. And this is where we'll find our candidate genes. And if we want to understand what those genes are doing, we can perform a gene ontology enrichment analysis. And yeah, we have a lot, and you guys can't see that. So I'm gonna zoom in to the top 25 and look at them a little bit more closely. So in terms of biological process, we found that protein phosphorylation, I'm sorry if you still can't see that, but innate immune response and defense response were our most significant gene ontology terms. And for molecular function, we saw that protein kinase activity was really popping out too. So now it was a matter of classifying up versus down regulated genes. And so these were some of our most significant genes that were shared by all 3 species. And here are just a few of our upregulated in terms of the good condition and downregulated in terms of the good condition. And in sake of time, I'm just going to focus on one. So if we look at RAB18, for instance, this codes for a dehydrin family protein. And something interesting about this, if we see expression of, or the presence of ABA, we see greater expression of RAB18. And something interesting about ABA is it facilitates tomato closure in response to drought. So this kind of allowed me to assume that furs with good needle retention induce tomato closure to conserve water due to dehydration. So that's just one conclusion I've drawn from this study. And just again, to reiterate, I did utilize a multi-assembler and we did see more upregulated genes and downregulated genes in the good condition versus bad condition. And again, RAB18 might suggest that these furs retain needles because they conserve water. That concludes the presentation. I want to thank everybody listed here. There's quite a few people who've worked on this project over the years. Thank you.
Only one question. Sorry.
We should talk first of all. And second, do you think the way these issues were collected and sampled are influencing your gene set? Do you think maybe there would be a more targeted way to elucidate gene discovery?
I mean, it's possible. I'm not super familiar with how it was collected because it was so long ago, but I know that we do have clones, and the distance between those clones being extracted is actually different for Fraser versus balsam and Canaan. I don't know if that would impact it. We had to remove some samples because our PCA plot was showing that some of the clones are actually clustering, and that was weird. So there's a little bit like we should probably look into in terms of sample collection, but yeah, I will talk to you.